This is funny because I think my presentation is just full of pictures, again, Gary. So Gary always says that I do nothing but make pretty pictures for the last 4 years, so hopefully I haven't disappointed you again. So a lot of the drive in microbial ecology recently has been really focused on understanding the human microbiome. And that's sort of led to the idea that we can manipulate the microbial communities living inside of us as a form of microbial medicine to treat certain human diseases. You may have heard about this in the form of fecal transplants, which I think kind of fits into the conference theme. But because microorganisms are essential to the functioning of all organisms on this planet, we can apply the same kind of ideas to the management of seagrasses. Of course, it's not that simple, as I'm sure a lot of you know. Microbial communities are incredibly diverse. Not only do they vary from organism to organism, but also across environmental gradients, as you've seen just in Matt's talk as well. So it means that even asking the simple question of can we use them to prevent and manage seagrass decline is an incredibly challenging question to answer, but it's not completely crazy. We have been using the association of plants and their microorganisms for our benefit for a long, long time. Classic examples of these would include the use of plants associations with mycorrhizal fungi, which allow the plants to acquire more phosphorus from the soil, or legumes symbiotic associations with nitrogen-fixing bacteria that allow them to acquire more nitrogen, and also the use of plants and their associations with microbes in various phytoremediation technologies, including design and implementation of artificial wetland systems. So we can apply these same ideas to seagrasses. And one of the areas that we could start thinking about this is what Matt's talk touched on as well, and that's the below ground environment, in particular when we're thinking about sediment health. And a lot of the time when we're thinking about sediment health, we are talking about sulphides that can accumulate to toxic concentrations in some sediments. And that's just because of the anoxic nature of a lot of seagrass and the majority of seagrass sediments, which means a lot of the microbial mineralization that's going on in these sediments is occurring through sulphate reduction. And that's obviously important to regenerate nutrients in the sediments, but under certain situations it can be bad if we have things like high organic matter in the sediments, if we have things like increasing in temperature, things that might reduce light to the seagrasses. A cumulative effect can mean that these sulphides can become problematic for the seagrasses and lead to large-scale losses. Of course, seagrasses are pretty hardy, as a lot of you know. They do have some adaptation to deal with the anoxic nature of these sediments and some of these sulphides, and one of them being these large air spaces throughout the tissues that allow that rapid diffusion of gases, and it can also allow the reoxidization of any sulphides that can enter into the below ground tissues. We can measure that internal oxygen concentration of seagrasses by installing ridiculous structures under the ocean that kids like to jump off on the weekend, even though we put signs to stay off. Here we use the scaffold structure to insert these oxygen microoptodes into the rhizomes of Posidonia sinuosa in this example, and then measured these concentrations over a day-night cycle. And obviously during the daytime you can see the plant tissues are quite aerated, and then when our light goes over the nighttime, some of these plants can actually become anoxic for several hours before the sunrise. And then of course again when the light is available again, these tissues are re-aerated. Actually, when these seagrasses are photosynthesizing and those tissues are aerated, some of that oxygen can leak out into the rhizosphere, which is what was just brought up before. And using, I think this is everybody's favorite seagrass at this conference, it seems like, as an example, Halophila ovalis. This is what this looks like for this seagrass. So this is a root of Halophila ovalis going through the sediment. The blue here is just the percent oxygen that's leaking out from these roots. There's kind of two things to notice from this oxygen leakage, and that is, one of the things is that this process is very spatially restricted. So that area there is just immediately kind of behind that root tip zone. The other thing to notice as well, at least for this species, and we've done it for Zostera as well, as these roots continue to grow through the sediment and then reach an age where they stop growing, that oxygen loss actually stops as well. And of course, because it's related to light availability, when you turn the lights off on these guys, this oxygen loss sometimes still goes on to some extent or completely ceases. This oxygen loss into the rhizosphere has been implicated in being important for sulphide detoxification in the rhizosphere. So here is a case example of using Zostera, where we have several root tips here leaking oxygen, and then you have a corresponding decrease in the sulphides in the rhizosphere around those root tips. So that sort of suggests that this O2 can actually protect at least those vulnerable growing root tips from the sulphides from entering into those areas. But when you look at older roots that have kind of stopped growing, you actually have a sort of opposite thing occur where you have an increase in sulphide production along these roots. So that sort of led us to start questioning what's going on with the specific bacteria around these root zones. And obviously focusing in on the bacteria that are involved in the sulfur cycling, including potential sulfate-reducing bacteria and sulphide-oxidizing bacteria. One group of sulphide-oxidizing bacteria that we were particularly interested in are the cable bacteria. These guys are quite cool because they are basically a filamentous bacteria that branch these oxic and anoxic microlayers where they connect the oxidization of sulphide in that anoxic layer with the reduction of oxygen in the oxic layer by moving these electrons from one end of their filament to the other. So they're basically skipping that classic redox ladder. If they were found hanging around seagrass roots, it would be obviously beneficial for the seagrasses because then you have an extra biological mechanism to potentially reduce those sulphides in the rhizosphere before they can enter into the root. So the title of my talk, we did go fishing for them, which actually stands for fluorescence in situ hybridization for anyone who doesn't know what that means. And it's basically using various DNA-based probes that have fluorescent dyes on them, and these probes you can use to target various bacterial populations and taxa that you might be interested in. And in this case, we were using various probes to target the cable bacteria, or our SOBs, and our sulfate-reducing bacteria, or the SRBs here. So to just give you an example of what we found using, again, Halophila ovalis, everybody's favorite seagrass, up here in the root hair zone of a root that is leaking oxygen, we actually do find quite a relatively high abundance of these sulphide oxidizing bacteria hanging around here. So that's what you can see, the purple in this image here. But when you move to a root that's no longer leaking oxygen, so those older roots that are probably in the stages of beginning to decay, you see very little, if any, of these cable bacteria in this zone. You obviously see a great increase in bacterial colonization in general, but you also see a great increase in the relative abundance of these sulfate-reducing bacteria, which are all the ones that are in yellow in this picture. See, Gary, lots of pictures. Yes. Okay. When we move into the area of the root where most of that oxygen loss is actually happening, we actually see very little bacterial colonization in general, and we really don't see any cable bacteria down there. But I guess that would make sense because that oxygen leaking out from that area would be oxidizing those sulphides around that area of the rhizosphere, so there's just not really any sulphides for them to potentially use. When we move instead to an older root that's no longer leaking oxygen from that area, again, we have a great increase in just microbial colonization in general. But again, we have a greater amount of these sulfate-reducing bacteria in this zone as well. So like I said, there's probably no cable bacteria in this area, but also because the sulphides in this zone would be less. So, to sort of bring that into a summary there, so the idea here is that oxygen leakage can protect seagrasses from sulphide by sort of two ways. There's the one idea of decreasing sulphide around the rhizosphere by, at least on those vulnerable growing root tips, but it can also function to decrease the abundance of the sulfate-reducing bacteria, the guys that are actually producing the sulphides to begin with, but also promote the occurrence of these cable bacteria, sulphide oxidizing bacteria. This means that our cable bacteria appear to be correlated with oxygen loss, at least for Halophila, ovalis, and Zostera, which really links that above ground environment and ability for a seagrass to obviously photosynthesize in the first place with the below ground microenvironment. It also seems to suggest that these guys are potentially key members of the seagrass microbiome, and I know they are starting to pop up in a lot of species around the place. But really now we need to test their function. Are they actually beneficial? Can we actually make that link to improving seagrass health? And hopefully then potentially if we can make that link, can we actually use them in things like restoration? And that kind of gives me a bit of a segue that goes on to, I think, some of what Gary was talking about with the seeds earlier today. At least for Posidonia australis, we've been trying to use, I think John's going to talk to you about it tomorrow, seeds of the Posidonia australis in restoration back in Perth. And I actually looked at some of these seedlings that I grew back in tanks, and we found all these cable bacteria all over these seedlings as well. So here, this green is actually the root surface of Posidonia australis seedlings, and the yellow in this time is all these filamentous cable bacteria. So that was kind of an exciting finding, because then it sort of leads us to think, well, if we can prove that they are beneficial to these seagrasses and start doing those sort of more manipulative experiments, it sort of suggests, is there potential for them, for us to use this in these restoration technologies as well? And yes, thanks to all these people, including receiving a travel grant to get here, which was very beneficial.
Thanks, Wanda, for a great talk. Any questions? Yeah, Gary's at the top.
Lovely pictures.
Like
I just love the pictures. But I have a little question about. So the cable bacteria don't occur where oxygen is leaking out.
Yes.
Or
Yeah.
where you can, is it where it's not leaking out, or where you're not measuring it, where you can't measure it because of low concentrations?
Yeah, that's true as well. It is like, so for measuring the oxygen from those roots, we were using oxygen planar optodes, which is obviously limited by the resolution of that technique as well. So it is possible that there are very tiny amounts of oxygen that are leaking out from that root hair zone that we're just not picking up. But yeah, where the majority of that oxygen is leaking out, I couldn't find any cable bacteria in that area. Yeah.
Being a non-microbiologist, you say it might help in the management maybe in future time. How do you emphasize that? How do you see that?
Well, that's what, yeah, I mean, I think this is what goes, what we're trying to kind of develop a lot, at least back in Perth, and I think a lot of people are, is really going from that correlation of all of these things to actually proving that these things are being beneficial. Yeah, I mean, those sort of experiments are the next steps, are the sort of things that I want to do. And those things are hard to do as well, like manipulating certain bacterial communities and populations. At least in this case, there's one target group that we can go for, which makes it somewhat simpler than trying to manipulate an entire microbial community. So it does give potential to start doing those sort of experiments, I guess, to tease that apart. Yeah. Plans for the future, maybe.
One more quick question. Oh, cool. Given unlimited. Oh, sorry.
No, this is
Oh, sorry, Casper.
like the sewing you in there, like the cable bacteria is actually classified as sulphide bacteria. They just run their genes and reverse over.
Yeah, their taxonomy seems to be all kind of, I think, in a state of flux at the moment, because there was that paper that was not, it was only published, I think, last year or something, that actually found that they're finding them in the freshwater environments as well as marine, and they're sort of separating and things like that. So, Yeah, I mean, this is the thing with a lot of molecular biology, it's only as good as our databases, really. So that's why, you know, those next steps are actually doing those experiments to try and prove that they're a beneficial thing to these seagrasses is really important. Yeah.
Thanks, Belinda.
Thanks, Matt.